All right, good news. The government has a continuing resolution till December 11th. So TRACO, we now have the green light. And later this week, I'll be sending emails to all the people who signed up to visit the cores and the tumor boards, and you'll get your assignments. And today our first speaker is Anton Simeonov. He got his PhD from the University of Southern California. He subsequently did a postdoctoral fellowship at the Scripps Institute, and then he came to the NIH, and currently he's the scientific director of NCAT's. His title, Challenges and Opportunities in Small Molecule Translational Research. Anton.
Thank you so much, Terry, and thank you all for coming in today. Yes, we're continuing our operations after having made all the plans for who's accepted and who is not. So let's see if we're going to be able to project again. All right, so what I would like to talk to you about today is how NCATS is addressing these problems in translation and what translation means for us and how we specifically work on small molecules to advance translation. So NCATS is one of the newest institutes and centers. Yes.
We're not viewing them in Frederick just yet. Nope.
Can you see them now?
Hang on one second. There we go.
So you see a slide titled NCAT's Mission, right?
Yes.
Yeah. Okay. So our mission is captured here in this long sentence: to catalyze the generation of innovative methods and technologies to enhance development, testing, and implementation of diagnostic and therapeutics across a wide range of human diseases and conditions. So it's a mouthful. What it means to indicate is there are many barriers that prevent you from going from a science paper, for example, describing a new pathway or new molecular target in disease, and an actual therapeutic intervention, whether it's through a small molecule drug or therapeutic antibody or diagnostic system. There are many, many hurdles that need to be overcome between the point of declaring victory, as in first author paper in Science, and actually helping patients through that discovery. It's a very lengthy, failure-prone process, extremely expensive, and what NCATS is trying to do is improve that process, increase efficiency, lower costs, and by doing so, help all boats rise, so to speak. So what are some of the translational problems? There are actually two kinds of problems in translational space: scientific in nature, organizational in nature. And the list is actually pretty long, but here are really the top problems that we're attempting to solve. There is really no good way to predict toxicology or toxicity of a chemical or drug candidate. A lot of drugs fail at that stage after having accrued huge expenses for development. There's still no good way to predict whether something will be toxic once it enters humans. So this is a huge problem, leads to a lot of attrition in development of both biologics and small molecule drugs. Same applies to predictive efficacy. You may have efficacy of your molecule against molecular target in a test tube, in a biochemical assay. Can you predict whether that molecule will be efficacious in a human to actually relieve symptoms? What are the models that are missing to make that prediction a little better? A lot of drugs fail at late-stage clinical trials because they don't meet the efficacy milestones for the disease. They work on mechanism, they do everything, they're not toxic, but they just don't move the needle. And there are many other problems in this space, including problems related to clinical trials, how to improve clinical trials, how to harmonize institutional review boards. All of these are little problems that are problems of every institute here at NIH, every drug development organization, and because they're so common to everybody, practically nobody is working on them, and that's what we at NCATS try to address. There are also organizational problems in translational space, data release, data transparency. People tend to guard their data because that's what is going to ensure their tenure approval or latest, greatest paper. IP management, incentives for team science, going again from the science paper to the therapeutic requires pretty strong team effort. You have to kind of drop your ego. You have to be comfortable being middle author on the paper. So how do you actually get incentives for being a good team player? Also, collaborative structures, public-private partnership models, we're attempting to change those because traditional ways to incentivize collaborations between academia and industry are not working very well. So all of these are problems of organizational nature. So it's pretty large space, and again, we're approaching it from disease agnostic standpoint. So to focus on small molecules, this is kind of a naive view of how you go about discovering a drug, right? You have presumably a molecular target. You actually have to create a test system to evaluate your drug candidates against that target or pathway. So you call that test system an assay. It is a test that is highly reproducible, relatively simple, and hopefully subject to automation. So you can run it many times over and not lose too much time on it. And then there's stages of testing of various chemicals, either libraries to discover initial chemical matter. So this is the process of high throughput screening. Or if you have a starting point, you can immediately start making chemical modification through medicinal chemistry to improve various properties such as affinity towards the target, to improve solubility, cell permeability, half-life in blood, all kinds of properties that then make that molecule a good candidate for testing in animal models and then ultimately in humans. So you can see the costs going up by increments of tenfold, typically. And of course, when you traverse the whole path, you're looking at probably more than $1 billion invested. So the science paper, again, is right around here, and somebody has to do all this and to do it more and more efficiently from now on if we want to actually reverse this trend of there being relatively few new drugs introduced to patients over the past couple of years or even more than a decade. So efficiency of drug discovery actually has been going down per unit of investment. So what are we trying to do at NCATS to improve that process? And I'm going to give you various examples from different steps of this process, how we address them through various programs. So we operate using the collaborative model. We have probably over 300 collaborations right now. So every problem we try to solve is not a self-invented problem that we solve internally and then move on. It is a problem that somebody else experiences, whether it's in infectious diseases, oncology, basic biology. So to demonstrate the solution, we actually have to have a collaborator who is expert in that particular disease or biology area. So in turn, to them, we provide the technology access and process improvement. So to discover small molecules, you have to set up various assays. And run various tests on small molecules, as I mentioned earlier. So these are the various assay types that we can run at NCATs. We can approach the system from a high-level phenotypic angle, looking at cell morphology. Various organelles can be stained and monitored for changes. We can focus on specific pathway by embedding reporters such as luciferase beta-lactamase, or we can drill even deeper and be very reductionist, looking at a specific protein and its interaction with the small molecule drug candidates, such as using enzymatic reaction readouts, protein-protein interactions, and so on. So we're equipped to do all this, and we run these tests in 1536 well plates. So this is really the cutting-edge technology that we adopted about 10 years ago, actually, and we've been perfecting it with time. And I'll mention what improvements we've made. Why do you need to go to such low volumes and such high density plates? It is to save time and reagents. Library that is, say, 1 million compounds or even more, if you run it in 96 well plates, you'll consume a lot of reagents, and it'll take quite a bit of time to actually execute the screen. In 1536, you can run things a lot more rapidly, and because of that, you can actually ask different questions as you run through these experiments, and I'll give you an example of how we changed the paradigm. So all this testing is effected in this automated system. You can think of the 3 orange arms as 3 technicians. They approach microtiter plate, scan its barcode, pick it up, and move it from workstation to workstation. And then we have various units that allow us to add reagents to these plates reproducibly at nanoliter aliquots, actually, to incubate cell-based assays in high humidity, 5% CO2, 37 degrees, so basically controlled environment. And we have the libraries of small molecules available on random access basis and all sorts of detectors to measure the outcome of these assays. So the system runs pretty much 24/7. We've modified it several times to essentially integrate latest and greatest plate readers, because that's really the technology that changes the fastest. We've run well over 150 full collection screens on over 500,000 compounds throughout the years. What did we change? What did we improve in this process? So I'll keep referring to this notion of improvement, right? How do we make sure that all boats rise? How do we improve the process? How do we increase quality, lower cost, and so on? So one thing that we thought about, again, a decade back when we were installing this equipment, was, can we actually run screening in a different way so that we can make it more efficient and informative? And one immediately obvious thing was, screening is traditionally run at single concentration. You test your drug candidate at 1 molarity, and you apply statistical cutoffs to the data, and you pick hits, so-called, and then you retest them, and a lot of them drop out because they turn out to be single-point artifacts. Wouldn't it be nice to actually test compounds in multiple doses, the way one would approach a pharmacological question? What is the potency of the molecule? Obviously, dose is important in not only basic biology, but in drug applications. You don't just take a drug at any dose. There is a recommended dose. So long story short, we actually worked for almost a year to design a new process to incorporate serial dilutions in this library preparation. So these different intensity colors, or different colors are different compounds, different intensities or different dilution points. You run this library at anywhere between 7 and 15 concentrations, spanning 4 orders of magnitude of molarities, from low nanomolar to double-digit or triple-digit micromolar. And in the end, you construct those response curves based on the screening data. We had to design specific plating processes to make this possible and efficient, and we actually had to write totally new code to develop browsers in informatics pipeline to calculate, say, 500,000 concentration response curves, 500,000 Hill equation fits to be performed automatically within, say, 20-something minute time span with minimal intervention. You cannot do this in Excel. You cannot do it in GraphPad Prism. So we had to design a new informatics process to do that. So you can generate, by doing so, pharmacological or toxicological actives with their potency, with their specific curve shape, whether it's the traditional shape or biphasic, whatever, we can actually distinguish all these for the entire library. And this is only made possible by using this automated system in very low assay volumes. That's the only way you can actually generate so many data points without breaking the bank. And then in the end, you dramatically increase reliability. False positives and false negatives decrease dramatically. And you can also compare your actives based on chemical similarity and derive structure-activity relationships from the primary screen. So we obviously developed this process, proved that it works, and we published it not just in summary form, but also providing source code, equipment programs, so that everybody can replicate it out there. So again, another common motif in this improvement in translational space, we don't hold anything back. We try to give it to the community because that's the only way to really improve the process for everybody, not just for us internally. So outreach is really huge deal for us. What is a screening assay? What is an automatable assay? Western blot is not a screening assay, for example, even though it's used many times a day on this campus and in every lab around the world. It is not a screening assay. So to teach people how to approach this, we actually worked with Eli Lilly. We were able to convince their lawyers to release the internal assay guidance document, which we then edited over the years, and now we have a pretty large editorial group, and it's now an ebook on the NCBI bookshelf with more and more chapters being added and about 20,000 or so hits every month and growing. So if you want to learn about assay development, assay artifacts, false positives, you name it, it's all there. And we're adding chapters on IPS-based screens, on testing of biologics, and so on. So it's actually a pretty good resource. So what is the space we operate in? We clearly don't want to compete with pharmaceutical companies on development of commonly needed drugs for, say, diabetes or other such indications. An area of unmet need where one really needs improvement in processes is the neglected diseases area. And NIH, as a public health organization, is obviously very interested in closing this gap. So there are about 7,000 diseases affecting humans, and probably no more than 500-600 of those, so 10%, have a therapeutic intervention associated with them. The rest are just sitting there, and doctors are trying to basically treat symptoms and not the disease. And there are actually two types of neglected diseases. The ones that are low prevalence, also called rare or orphan diseases. So these are usually genetic diseases affecting very, very few patients worldwide. Some of them are ultra rare, affecting probably 500 patients worldwide, or even less in some cases. And then there's the other kind where a lot of people suffer from these, and they're usually infectious diseases in kind, but the population resides in third world developing countries and cannot pay for the medicine. So you're looking at some very traditional diseases like malaria, but also schistosomiasis, leishmaniasis, and others that are not making the headlines every day. So clearly two areas of neglect or unmet need, and I'll give you an example of each area, how we actually address gaps in translational development. So on the side of early discovery, we actually worked a couple of years ago on schistosomiasis. It's a pretty nasty infection caused by this worm shown here in this blown up image. The worm actually penetrates the skin of people who bathe in rivers or lakes and ultimately makes its way into the liver and resides there for a pretty long time. It can actually survive in the human host for probably 20, 30 years and go undetected and gradually grow inside the liver, and these kids look like they're pregnant, but that's because their livers are 7 to 8 times their normal size. So huge number of infected individuals, mostly sub-Saharan Africa, parts of China, up until recently Japan as well, but very few deaths, relatively speaking. So it's not as famous of a disease as malaria or related diseases like TB that make the headlines, but morbidity is actually pretty big. People can't really walk to go to work. They just sit there and slowly expire. So interestingly, because the parasites can remain in the human host for so long, they clearly must have developed some detoxification mechanisms against innate immune system reactive oxygen species. So this is really the development that caused us to run screen for schistosomiasis. This collaborator, David Williams, he's a faculty at Rush University in Chicago, discovered this redox pathway and published the pathway and the target. Long story short, turns out that unlike humans, where detoxification of reactive oxygen species can be performed by two enzymes, thyrodoxine reductase and glutathione reductase, in schisto you have only one enzyme kind of converged both thyrodoxine activities and glutathione reductase activities in this thyrodoxine glutathione reductase. So this enzyme naturally provides a bottleneck in the parasite's detoxification mechanism and allows it to be used as a drug target. So we set up a screen using coupled enzyme system following actually NADPH fluorescence in real time, followed the screen with various secondary assays to determine what the hits were doing. And out of the screen of a relatively small collection back then, we got various hits, different chemotypes, and interestingly, compound that was not the most potent against the enzyme. So you see IC50 here of over 1.5, close to 2, as opposed to others that are nanomolar. This turned out to be the compound with the strongest effect on cultured worms. So we were lucky to have, through this collaboration, the model that allowed us to treat worms in media with the compounds and literally look for morphological changes in death. So this was then followed by testing this compound in vivo. So this compound is called furoxan. It's actually a known NO donor. It is not an approved drug, but it's being tested for various other indications. So mice can be infected with schistoworms, and that can serve as a natural, simple, relatively simple model for the infection. And what this set of images shows is untreated liver full of eggs, schisto eggs, very large size. And then upon treatment with the furoxan molecule, at different stages of worm development, you can see that invariably the worm burden is brought down to almost the 0 level, and also the livers are brought back to a more normal size and practically free of eggs. The compound was also active against different species or isoforms of schisto. So was broadly acting, unlike existing therapy that is only active against adult worms. So this was an interesting story. Of course, you see where it was published, generated all kinds of initial publicity back then when we ran the study. But most importantly, it was seen as a good example of how translation can address these areas of unmet need, that is, neglected tropical infections in that case. And we were able to take this to the bank in almost literal sense. Congress actually authorized the creation of this Therapeutics for Rare and Neglected Diseases program, which is now part of NCATS, to deal specifically with de-risking drug candidates, whether biologics or therapeutic oligos or small molecules, for the purpose of treating rare and neglected diseases. So the TREND program is still part of NCATS, and it handles drug candidates that were initially discovered by either academicians or small biotechs, but whose development has been stalled for either business reasons or company running out of money or company not having the expertise at a particular stage of development. That's where our program comes into play to just help with a couple of steps of this development so that the project can cross the valley of death, so-called. So to give you an example of what we've done for another rare disease within the TREND program, this is, as you can see, pretty well-known sickle cell disease. Rare disease was the first one for which the genetic cause was discovered back in the day, and there's still no good treatment for it. So the small company, SRX, had discovered this very small and arguably ugly-looking chemical, hydroxymethylfurfural, as being able to bind to sickle hemoglobin and shift its oxygen binding curve, thereby reducing sickling. So this is all good, but it's a proof of concept in vitro, and no investor was willing to put money into the company because the molecule looked so ugly. It has aldehyde, small, potentially reactive, so there were concerns about toxicity, formulation, and the company was literally stuck. They had, again, good evidence for efficacy, but they could not move any further. So this is, again, this valley of death, this area of quote-unquote boring work, where somebody had to help the company with toxicology and formulation, something that does not lead to a science paper, something that is not really the final clinical trial, but it's a required unit of work to help that program become attractive from a commercial standpoint. And that's what our team did. We helped them run animal talks, worked on CMC, which stands for Chemistry, Manufacturing, Quality Controls, and also helped with some regulatory aspects, interactions with FDA. And long story short, the company was acquired by Baxter, their blood division, and now Baxter is actually bankrolling the development of this molecule and actually sending it to clinic and so on. So this is an example where we're acting pretty much as a catalyst. We took the project for very little time, just helping the company with 2-3 steps along this drug development path, and now they're off on their own, and we're done, and we can move on to helping another company or academician. So this is an example of how we work in this later stage of drug development. I want to shift gears and talk a little bit about different approach to discover drugs for new indications, which is to repurpose existing drugs. So the traditional process requires one to run screens of large libraries and then spend a lot of time optimizing the molecules, doing all this preclinical development, and then hopefully gain an FDA approval. What if one can start with existing drugs which have been shown to be non-toxic in humans, safe for use in humans, and simply test them for a new therapeutic hypothesis, maybe change their formulation or dosage a little bit, and gain approval for this new indication within about 1 to 2 years, keeping a lot of steps and a lot of costs. So this is the concept of drug repurposing, and there are several thousand such drugs. So to actually enable this process, one needs, obviously, screening system, some therapeutic hypothesis, but one also needs the drugs collection. And so for us, the first question was, how many drugs are out there? And you may say, well, can't you look them up? Isn't there a website of approved drugs? Answer is no. Actually, even FDA doesn't have a full-blown website that tells you what is approved, what is investigational, and all that, let alone the rest of the world. So our informatics team spent about 4 years going through websites, databases, disambiguating spellings of different versions of the same drug, getting all the way to Cas numbers and an enantiomer level information, and building this database and browser, which was published a couple of years ago. And then we actually embarked on building the physical collection of about 4,000 drugs, which we're still building and we're using for repurposing screens. We're also working in this other area, which is sort of companion area, drug repurposing, which is to look for novel drug combinations. So this is not a new concept. It is well known from areas of AIDS treatment, the HIV cocktails of 2-3 drugs. They've been approved and have shown to be pretty efficacious. Why would you want to have drug combinations? You may gain advantages in terms of efficacy, that is improved efficacy, or you can also lower the dose of each individual drug to lower the target-related toxicities. So all kinds of opportunities there. You can also use combination studies to as a hypothesis building exercise to understand why two therapeutic agents or small molecule agents act synergistically. Basically, it gives you an entry to systems biology type investigations. So how do you go about setting this up as a high efficiency, high speed exercise of finding novel drug-drug combinations? So we worked on this. Again, this is a platform technology that we developed the past couple of years. So to do this, you need a library. As I mentioned earlier, we're building the largest such collection of approved drugs and investigational agents. We also need highly effective way to mix these drugs in pairwise fashion and do it very rapidly, reproducibly, and comprehensively, and also an automated data analysis method. So I'm not going to go into this in huge detail, but the way we're approaching this is we first generate single agent results to guide us as to the approximate potency of these drugs in that particular assay. Then we stamp using acoustic dispense, which is literally like a printing technique, spotting technique, 6x6 matrices of 6 concentrations of drug A against 6 concentrations of drug B. So these are still binary combinations. We don't combine more than 2 drugs at a time. So for every combination, we get 36 data points spanning certain molarity range, which is dictated by the potencies of the individual agents. We don't want to overshoot. We don't want to undershoot. That's why it's important to have a system to plate these compounds in custom concentration manner. This is a huge deal. It cannot be done effectively using traditional pipetting equipment. You will run out of the delivery range. You'll spend a lot of time actually making serial intermediate dilutions, and errors will propagate. So this acoustic dispense technology allows us to do it extremely rapidly and efficiently. So combining, for example, let's just say cisplatin, somebody's favorite oncology agent, with the entire pharmacopoeia, 3,000, 4,000 drugs. So think 4,000 combinations. Each combination having 36 data points. It takes us about 3.5 hours to stamp those plates, so we can combine that process with preparing cells, either using primary patient cells, for example, in leukemia cases, one can actually do that, or tumor line, whatever you're studying, and run this assay on freshly prepared compounds and get that data within literally an overnight period. We have automated data analysis method to build these heat maps and calculate various indices of synergism. And then we follow this primary screen by arraying the hits with 10 by 10 concentration combinations. That way we get more dense coverage of actual curves and can verify the synergism. So not to bore you with details, you can look up the process in this paper. It was actually initially funded through collaboration with NCI, with Lou Stout's group. And again, within this paper, we not only presented the results from this first screen, but also the entire data browser and acoustic dispenser source code. So again, we didn't keep anything proprietary. We're giving everything to the world so that others can replicate these methods. This is the only way to advance translation in the truest sense, is to give data methods to others. So I mentioned predictive toxicology as one of the main translational problems in developing small molecule and large molecule drugs. So here's the plot that kind of gives you further evidence for why this is not a solved problem yet. So about a third of all drug failures are due to preclinical or clinically manifested toxicity issue. Notice how, for example, pharmacokinetics and bioavailability has been largely solved as a drug development problem. Toxicity has not. So this is a pretty serious impetus for one to try and work on better methods to predict toxicity of a chemical drug candidate. The other reason for wanting to work in this area is actually environmental reason. So there are over 80,000 chemicals that exist in commerce, and for most of them, probably 79,000 of them, there is practically no toxicological data against animal, human, anything. Why? Because there is no mandate to generate such data, and because it's extremely expensive to generate such data, because you have to use many rats and follow them for many years and so on. So all these chemicals are around us. We get exposed to them, bisphenol A, you name it, and there is very little data to inform EPA and the general public as to dangers associated with them. Also, there is enormous push to reduce use of animals in lab research. So how do we approach this problem? You can think of the attempt as trying to deconstruct the human or the rat into component pathways and test the chemicals using relatively cheap and scalable biochemical cell-based assays to determine their effects on these pathways, and then by building this very large database to attempt to reconstruct on systems biology level the effect of these chemicals in vivo. So this is the ambitious program we're started a couple of years ago. It's called TOX21, stands for Toxicology in the 21st Century. It's actually a collaboration between NCAT's, NIHS, the National Toxicology Program, FDA, Environmental Protection Agency. And the goal really is to begin to identify patterns of compound-induced biological responses in assay systems that have relevance to toxicity outcomes. Think estrogen receptor disruption, membrane potential modulators, inducers of DNA damage response, and so on. The intermediate goal being to prioritize compounds for more extensive toxicological evaluation and ultimately to develop predictive models for biological response in humans. By doing so, we hope to minimize animal testing and to improve predictive power of these early preclinical development activities so the compounds can be flagged as being potentially dangerous to humans before they actually get to either clinical trials or release into the environment. So to do this, we actually had to develop a whole pipeline of testing, and it started with assembling a collection of about 10,000 chemicals, and the various chemicals were contributed by the different agencies based on their long-standing interests and previous programs. There was deliberately overlap between the proposed molecules because we wanted to evaluate inter-vendor differences in the behavior of these chemicals, because depending on where you source some of these chemicals, they will have different impurity signature, and you actually want to know what the different versions do in the assay, because that's really the more realistic representation of what the chemical will do when released in the environment, for example. We included all sorts of positive controls, failed drugs, also the drugs collection that we're assembling for drug repurposing. And this library was actually plated as a triplicate set. That is, 3 separate instances of the library were created with the compounds moved around the plate with each replicate to minimize positional effects from screening. We included a lot of internal controls, 88 single-source compounds in duplicate on each plate to follow systematic trends across screening. And we tested every molecule as 15-point dose response to generate really dense data coverage. So we've been doing this screening for the past few years, and this is an example of the output. One of the first screens, given its importance, was the estrogen receptor alpha, using various readouts, luciferase reporter, beta-lactamase reporter, using ligand binding domain only or full-length receptors. So we wanted to really study the system in detail to figure out what we can get away with going forward, because obviously the more versions of one pathway you run or one receptor, the more expensive the whole operation. So this being one of the first projects, we wanted to do it exhaustively and then figure out what is sort of possible to skip or simplify. So so this is the initial review of the data and initial analysis. And of course, all the data are made public, but we don't even begin to pretend that we can make sense of this data. This is really big data, and we've published actually over 30 nuclear receptor signaling screens and stress pathway screens, and basically we turn to crowdsourcing to sort of get wisdom from all this data. So what we did was we provided about 50 million data points, all individual data points in PubChem to the entire community with no restrictions. We also provided the structures of these 10,000 chemicals that were tested. So for every chemical, you knew its name, CAS number, structure, and then all the data down to single activity data point. And the goal was for people to submit predictive models to predict the response of a new chemical based on its structure in that particular assay. So it's not the ambitious goal of predicting response in humans yet. It's to predict response in the same assay, but it was still pretty ambitious for the state of the field. So we had a tremendous response, actually, from all over the world. We used 300 new compounds that we had tested in the assay for which we had not provided the data to the community to score these models. And it's interesting to look at the list of winners and concentrate probably just on the city and country here. you will see no single winner from the United States. So I'll let this sink in. This is an excellent argument for why it's good to share your data. You just don't know who's going to make the best sense out of it. Very small countries, very small universities, all these winning teams from Austria, Hungary, and other places. So it's a lesson learned. It's good to share your data, run some initial analysis, publish the initial findings, but do share your data because that's really the best way to advance the field. We should all learn to be more selfless in this regard. So we intend to use this crowdsourcing model for other problems going forward, both on the tox side and drug efficacy side. So in this sort of first and second phase of Tox21, we've built a platform to run very large-scale testing of thousands of chemicals, hopefully graduating to tens of thousands of chemicals across multiple cell-based assays. All these data have been made public, along with protocols, publications describing the methodology. We're also engaging overseas partner agencies and institutes to recruit better assays. And we readily recognize the limitations of this approach. We're still just treating cells in two-dimensional culture for only a couple of days and taking very simple readouts. We intend to increase the role of or usage of IPS in future testing. I'll tell you a little bit about that in a second. We're also making efforts to increase the sophistication of data gathering by setting up high-content screens, looking at multiple pathways simultaneously, taking snapshots of the whole transcriptome of a cell as a result of treatment with a chemical. We're working on next-generation transcriptional profiling technologies that would allow us to do that cheaply and efficiently. And we're also working on ways to embed metabolic processing into these assays, because right now they're very simple. You add chemical to a cell, there is no liver cell, there is no metabolism. We need to improve on that as well. But as far as setting up the initial technology, we're pretty much done with the robotics and informatics part and compound sourcing and QC part. So I mentioned IPS technologies. We're also making efforts in this direction as part of a collaboration with the NIH Common Fund, the Office of the NIH Director, to set up a stem cell technology facility at NCAT to work on common technological hurdles in this field, specifically problems that prevent one from using IPS-derived differentiated cells for cell therapy. And really the two main problems in this area is lack of good reference methods to characterize cells, whether you've actually made fully differentiated, mature cell out of your IPS culture. So we'll be working on setting these standards, developing databases of cellular signatures to inform the community as to the status of their cells as they process them through differentiation protocols. The other big problem is improvement of the protocols themselves. Right now, there are a lot of protocols to make IPS cells, to expand them, to differentiate them. They're hardly reproducible, and for sure, they're extremely expensive and non-scalable. So what we're going to be doing is run high-throughput screens to find small molecule drugs or novel molecules to replace expensive protein factors and media components to improve the efficiency of reprogramming, potentially the speed of reprogramming, and definitely to lower the cost and improve reproducibility. And along the way, we'll be generating IPS-derived models of rare diseases, and also we'll be using IPS-derived terminally differentiated cells in toxicity evaluations. In this case, looking at cells differentiated to the same final lineage, but coming from different individuals, and really entering this field of toxicogenomics in this case. And again, as before, we're going to run these projects as collaborations and make all data, methods, and compounds publicly available to everybody out there to use in their research. So just to give you an example of how big this problem is, we're collaborating with investigator at the National Eye Institute here, Kapil Bharti, who's working on a protocol for actual cell therapy, the only one on the main campus of NIH, to go from patient-derived cells, patients with age-related macular degeneration, to convert them to iPS cells, to expand them, and then to differentiate them to retinal pigmental epithelial cells and return them to the patient's retinal area in the form of specific injection of a scaffold and the cells implanted in it. So this is the protocol to make RPEs. It's literally a 3-month protocol. And it has many media changes, many manual intervention steps, and it's fairly expensive and hard to scale up and reproduce. So what we intend to do is take this protocol and dissect it into literally dozens of screens, running small molecule screens at, say, week 1, week 2, week 1, week 2, this step, and so on, arriving at this matrix of data to try to find small molecules that will improve efficiency at any of those steps, or maybe all of them, or to just replace an expensive component. So you notice it's actually a pretty complex problem to attempt to solve. So it'll be done through actually a multiplicity of screens, not just one screen. Last thing I'd like to mention is something that we're doing on the extramural side of NCATS through a grant program, which relates to this question of improvement of predictive models. So this is the tissue chip program, which is designed to generate this in vitro platform that recapitulates major components of human physiology in microfluidic chip or cartridge format. That is to have liver on the chip, lung on the chip, gut on the chip, and so on, demonstrate their physiology, that these systems are alive and performing what they're supposed to perform as far as physiology, and then to begin to connect them to each other to take it to the next stage. So the goal is to have tissue be viable in these systems for at least 4 weeks, to have these be modular and reconfigurable so they can be connected, and in the end to have some assurance for community-wide access. Again, I'm not going to go into detail as to how this is realized. One has to account for all these elements of complexity when one builds a human on a chip. The cells have to be the right kind. Obviously, some of them come from primary cells, others come from iPS cells, and so on. One needs to take care of a scaffold, structure, patterning, ensuring perfusion, innervation, and then, of course, having some sort of readout. How do you measure that your gut on the chip is working? What do you actually measure? So all of this has been going on for a while. It's actually a collaboration with DARPA, the Defense Advanced Research Project Agency. Program is about halfway through. The individual organs have all been generated and validated. And now the program is entering this phase of integration or connecting different organs. So, for example, having gut on the chip, with liver on the chip, with kidney on the chip, feeding a liver toxic drug and actually demonstrating the drug is metabolized and that it exhibits, for example, liver tox effects. So this would be the use of this platform. Last slide here, you can learn more about it from the website. There's videos and all kinds of details for the various organs. And it's actually a pretty fascinating system. One grantee was even able to recapitulate female reproductive system with the 28 cycle on the chip. For ethics reasons, they cannot use human cells, so it was actually a mouse system designed to mimic the human biology with the 28-day cycle. I'll leave it at that. There's plenty of information there. So the hope is that within about a year or two, we'll start seeing convergence of all these technologies where the robotic screens, which can address tens of thousands of compounds, can produce various hits which can then be fed into organs on a chip or human on a chip to both better understand the mechanism of action of these hits from the cell screens and to validate their in vivo effect, whether it's toxic effect or drug efficacy type effect. So we're eagerly awaiting the productizing of all these various organs and physiological systems, and there are actually about half a dozen startup companies that have been spun off from the various academic institutions that worked on the various organs. I'll stop here. Thank you for your attention and be happy to answer questions.
So what type of cancer projects are you doing now?
So the combination screening platform was developed in collaboration with NCI. They've sent us probably about 15 different combination screens to work on with various investigators, Lou Stout, Lee Hellman, Javed Khan, various models, really. A lot of the readouts revolve around cell death and viability because they're relatively simple, but we're working on other readouts as well. We're also trying to combine immunotoxin with small molecule drugs. It's not as simple as combining two small molecule drugs because of different formulation requirements for, say, therapeutic antibody or protein immunotoxin, but we're working on that as well. So this is really where drug-drug combination platform has taken off, mostly oncology projects. We're also starting some infectious disease projects with the drug-drug combination platform. We have also a lot of efforts in the area of rare tumors, pediatric cancers, probably over 80 different projects in the cancer field, both target-based and phenotypic.
Okay, Anton, thank you.
Thank you. All right. Let me switch off. Okay.
Okay, one additional announcement is next Monday is Columbus Day, and that is a government holiday. So next week we'll be having TRACO on Tuesday at 4 to 6 instead of Monday. So our next speaker is Sonia Jakowlew, and she got her PhD at Rutgers University. Subsequently, she did a postdoctoral training in France and came back to the NIH and started working on transforming growth factor beta. She's now in the cancer training branch. Her title, Transforming Growth Factor Beta and Lung Tumorigenesis. Sonia.
Okay. Thank you, Terry, for inviting me to this lecture series. Well, today I'm going to be talking about a growth factor, transforming growth factor beta, in a very serious cancer process involving lung tumorigenesis. And my seminar will include a brief background history about TGF-beta and lung tumorigenesis, lung cancer. And then I'll be presenting some actual experimental data from my laboratory. So, lung cancer has become the most common cause of cancer death among both men and women in this country. Last year, there were over 224,000 new cases of lung cancer that were diagnosed among both men and women in this country. In addition, there were over 159,000 new deaths due to this horrendous disease. Most cases now occur in former smokers because lung cancer, it takes a very long time to develop, and you usually don't see this until you've been smoking for some years or have quit. And despite our aggressive anti-smoking campaign efforts and the various new therapies and treatments for lung cancer, the 5-year survival rate for this cancer is still less than 15%. So transforming growth factor beta, or TGF-beta for short, is a multifunctional regulator of cell growth. It's a potent inhibitor of most normal epithelial cell proliferation and shows widespread tissue expression, particularly in mammals. TGF-beta has been shown to play a very pivotal role in maintaining epithelial homeostasis, and it's been shown to be associated with various types of cancer, including lung cancer. And most importantly, it shows context-dependent inhibition or stimulation of cell proliferation and neoplastic transformation, depending on the environment. So TGF-beta might be an attractive candidate for new therapeutic intervention approaches in lung cancer. So to understand TGF-beta, I have to take you back to the beginning. And TGF-beta has its roots in another growth factor called sarcoma growth factor. Sarcoma growth factor was a polypeptide that was demonstrated to be secreted by Maloney-Murine sarcoma virus transformed mouse fibroblasts that could stimulate normal rat fibroblasts to form colonies of self agar. And this was the transformation assay that was used to identify this growth factor by Joe DeLarco and George Todaro in the late 70s. Following the discovery of sarcoma growth factor, 2 classes of transforming growth factors were isolated from MSV transformed cells. One class was shown to be able to compete with epidermal growth factor, or EGF, for receptor binding. This was called TGF-alpha. A second class was found that did not compete for EGF binding, but could display colony-forming activity that was enhanced by EGF, and this was called TGF-beta. So sarcoma growth factor is really made up of at least 2 different growth factors, TGF-alpha and TGF-beta. And this was reported by Roberts and Sporn in a paper in 1982. Following the discovery of TGF-beta, the purification of TGF-beta was demonstrated from at least 3 different sources in 1983, from human platelets and placenta, as well as bovine kidney. And this was discovered by 3 investigators at the NCI. To give you some idea about the purification that was involved in purifying TGF-beta, typically 100 grams of bovine kidney obtained from the slaughterhouse was extracted with 8 liters of acid ethanol for several hours, then centrifuged, and then precipitated overnight with ether ethanol. The next morning, the precipitate was dissolved in acetic acid and then applied to 80-liter biogel P60 columns for purification. 1-liter fractions were collected, and then these were lyophilized and redissolved for further chromatography purification. So a final yield from about 100 grams yielded 6 micrograms of TGF-beta. Now, to give you some idea of the extent of the enormity of this purification, these are typical P60 columns, and the pink arrow shows a 55-gallon drum. So this was massive when they were trying to purify enough TGF-beta to work with. So the assay for TGF-beta was typically the growth of normal rat kidney cells in soft agar. So to do this, a mixture of media, serum, NRK cells, EGF, and sample were applied to an agar plate and allowed to incubate at 37 degrees for a week. The plates were then stained and colonies were counted with an Omnicon image analysis system, which is shown at the bottom of the slide. So if no TGF-beta was present in the samples, no colonies would grow and nothing would be counted. If TGF-beta is present in the sample, the colonies would form and they would be counted if they were above a certain size threshold. So shown here is the final HPLC purification of TGF-beta after all this purification measures. And TGF-beta migrates as a, see if we can get this to work. Okay, as a 25,000 molecular weight single band on a polyacrylamide gel. So, Eureka, TGF-beta is born at the NCI, and Drs. Michael Sporn and Nita Roberts are credited with its discovery and purification. Following the purification of TGF-beta, the amino acid sequence of TGF-beta was developed, and it was shown that TGF-beta actually exists as a pre-pro molecule consisting of a single peptide at the N-terminus, a middle latency associated peptide, also called the LAP, and a mature TGF-beta consisting of 112 amino acids at the C-terminus. The LAP is very necessary for this protein to develop into the proper configuration conformation for activity. Now, in the early 90s, the crystal structure of TGF-beta, and I should say this is TGF-beta 1, was developed, and TGF-beta was shown to be a dimer composed of 2 identical monomeric chains held together by interchain disulfide bond, which formed a hydrophobic pocket in the middle between the 2 monomers. TGF-beta is actually a member of what is now called the TGF-beta superfamily. In addition to TGF-beta, which consists of 3 isoforms, TGF-beta 1, 2, and 3, in mammals, there are 2 additional TGF-betas that have been identified, TGF-beta 4 in the chicken and TGF-beta 5 in xenopus. And TGF-beta 4 and 5 are thought to be homologs for the mammalian TGF-beta 1 isoform. We also have the bone morphogenetic protein and growth differentiation factors, which are very numerous, as well as the activins and inhibins in this superfamily. Now, shown in this cartoon is the basic structure of TGF-beta. It's a 25,000 molecular weight disulfide bonded homodimer. There are 3 highly homologous isoforms, TGF-beta 1, 2, and 3 in the mammals. Principal sources are platelets, bone, and spleen, but it's virtually found in every tissue in the body. Most cells express TGF-beta and its receptors, including normal cells as well as tumor cells. TGF-beta is usually secreted in what is called a latent inactive form, which has to be activated before TGF-beta can exhibit its properties. And the superfamilies of TGF-beta can contains not only the TGF-beta isoforms, but the activins, inhibins, BMPs, and growth differentiation factors. So it's quite a large family. Now, the TGF-beta superfamily, its members have been shown to play central control in many biological processes, including development, immune system function, reproduction, angiogenesis, aging, tissue repair and response to injury, metabolic regulation, and proliferative homeostasis. So it's involved in a whole variety of biological processes. Now, there are major biological responses that are regulated by TGF-beta. TGF-beta can inhibit proliferation under certain circumstances. It can also regulate apoptosis, differentiation, and the immune cell function. Under certain circumstances, it can be stimulative of the accumulation of extracellular matrix, and it can promote chemotaxis of certain cells. Now, the model for TGF-beta signaling pathway has been developed over several years, and it starts on the upper left. The TGF-beta ligand, and this may be TGF-beta, may be the BMPs, may be activin inhibin, binds to the type 2 receptor, shown in pink, which is constitutively phosphorylated. This complex then recruits and binds the type 1 TGF-beta receptor, shown in pink, to form a supercomplex, and the type 1 receptor transphosphorylates the type 2 receptor transphosphorylates the type 1 receptor. Now, as a complex, it is now ready to bind and interact with another set of proteins called the SMADs. And depending on whether you have activin, TGF-beta, BMP, or TGF, it'll bind to a different set of SMAD proteins. Now, the SMAD proteins that are involved with actual TGF-beta ligand are SMAD2 and SMAD3. Those involved with the BMPs are SMAD1, 5, and 8. So these are actual distinct proteins. And what they do is when they bind to the complex, they are then, these SMADs are then phosphorylated. And after phosphorylation, they're able to bind another SMAD protein called SMAD4, which is called the common SMAD. Now, SMAD4 has very unique properties. It can translocate into the nucleus. So it carries a signal into the nucleus where it can affect regulation of specific transcription factors. Now, this whole thing can be short-circuited by inhibitory SMADs. Whoops, sorry. Don't know how that happened. Inhibitory SMADs, SMAD6 and SMAD7, which can act to just negate the whole system, so there's no downstream signaling, which often happens in cases. Now, clinically, TGF-beta has been shown to be a tumor suppressor. It's been shown that there are germline mutations in specific TGF-beta pathway components that cause familial predisposition to cancer, such as SMAD4 has been shown to be heavily mutated in a condition called juvenile polyposis syndrome. In addition, several TGF pathway components have been shown to be either deleted or somatically mutated in several human cancers, such as the type 2 TGF-beta receptor in human non-polyposis colorectal cancer, and SMAD4 has been shown to be heavily deleted in human pancreatic cancer. Thirdly, reduced expression of several TGF-beta signaling pathway components have been shown to occur, as well as overexpression of endogenous pathway inhibitors have been shown to be associated with tumorigenesis. This includes the type 1 and type 2 TGF-beta receptors, as well as the inhibitory SMAD protein and the co-repressor called SCI. Now, clinically, TGF-beta has been also shown to be a tumor promoter. TGF-beta has been shown to be elevated in many types of advanced human tumors and to correlate with metastasis and poor prognosis. And this has been shown in a wide variety of different tissues, including lung. And on the right, lower right slide, is shown a prostatic adenocarcinoma that has been stained with TGF-beta-1 antibodies, specifically for TGF-beta-1. And you can see the brown staining shown by the black arrow, and TGF-beta seems to sit at the interface between the tumor and the microenvironment. Now, if you look at the light arrow at the top, you don't see any staining with TGF-beta-1 because this is normal tissue. So what's going on and why is this so confusing? Is TGF-beta in carcinogenesis, is it really a hero or is it a villain? Well, it turns out to be both. TGF-beta can be a proximal inhibitor of the malignant phenotype, but it can also be a potent growth inhibitor and tumor suppressor, and it can also be a pro-metastatic factor. And everything depends on the context and the environment that it is put in. So, unifying hypothesis has been developed, which tries to explain how TGF-beta switches from being a tumor suppressor gene to a pro-oncogenic factor as cancer and tumorigenesis progresses. So, in the normal epithelium, the tumor suppressor activities of TGF-beta predominate. But as changes occur in the genetic and epigenetic context as tumorigenesis progresses, TGF-beta loses responsiveness and TGF-beta expression and activation increases. So when you get more towards the invasive metastatic cancer, the pro-oncogenic activities predominate over the tumor suppressor activities, and you have pro-oncogenic phenomena. So in addition to the TGF-beta SMAD-dependent pathways that I just spoke to you about, there are other SMAD-independent pathways that work at the same time in both normal and tumor cells. And this includes such pathways as the MAP kinase pathway, the RAS pathway, the Rho, and the protein phosphatase 2A pathway. Now, we're particularly interested in the RAS MAP kinase pathway because the KRAS proto-oncogene has been studied in lung cancer, and KRAS shows an activational mutation in at least 25% of human lung adenocarcinomas. It's been shown that mutation of even one allele of KRAS increases the appearance of lung lesions, and it's been shown that there is tremendous crosstalk between SMAD-dependent pathway and the RAS-MAC signaling pathway. Fourthly, activation of the RAS pathway can modulate how TGF-beta-1 signaling occurs through the SMAD pathway. And finally, in vitro studies have shown that TGF-beta-1 dominates over the mitogenic effects of RAS, but that activated RAS can override the antiproliferative effect of TGF-beta-1. So it seems like TGF-beta-1 and KRAS can have an interplay in lung cancer. So we decided to look at the tumor suppressor and tumor promoter activity of TGF-beta, and up at the top, on the top left, is a normal situation where if everything proceeds normally in normal cells, TGF-beta acts as a tumor suppressor. But in tumor cells, if you have such activity as that decreased expression of the type 2 TGF-beta receptor, or hyperactivated MAP kinase with activated RAS, or decreased levels or activities of any of the SMAD proteins, or compromised effector function in any of the suppressor arms of the pathway, you might have a situation where you have tumor promotion. So the broad goal of my laboratory was to determine the role of transforming growth factor beta in the development and malignant transformation of lung epithelial cells. A very modest goal, okay? So this work was done in the epithelial carcinogenesis section of the Cell and Cancer Biology branch of CCR. So we had three objectives. First, to examine the effect of TGF-beta1 deletion and KRAS mutation alone and in combination on lung tumor incidence and pathology. Then to determine the early events in the development of lung lesions and how they progress onto tumors, carcinomas. And thirdly, to identify potential signal transduction pathway changes that may occur with advancing tumorigenesis. So for this study, we employed 4 mouse models: the AJ mouse, the TGF-beta1 heterozygous mouse in both the C57 Black 6 and AJ Black 6 backgrounds, and the TGF-beta1 heterozygous KRAS latent activatable mouse, which we obtained from Tyler Jackson at MIT. We asked 2 questions. Does lung tumorigenesis affect the TGF-beta signaling pathway? And the reciprocal question, does the TGF-beta signaling pathway affect lung tumorigenesis? So to answer these questions, we first utilized the AJ mouse model. The AJ mouse model is very susceptible to chemically induced lung tumors with such chemicals as ethyl carbamate. The lung tumors that develop in a very time-dependent manner and progress through the stages of hyperplasia, adenoma, and carcinoma. The carcinomas that develop have been shown to be histologically similar to human lung adenocarcinomas. And very importantly, the same molecular mutations occur in both human and mouse lung tumors, such as overexpression of RAS and the loss of P53. So to give you a little bit of background about ethyl carbamate metabolism, ethyl carbamate can be metabolized through 2 pathways. First is the detoxification pathway on the left, in which an esterase converts the molecule into harmless ethanol, CO2, and ammonia. There's also a bioactivation pathway that employs CYP2E1 in the lung and reduces ethyl carbamate to vinyl carbamate and a vinyl carbamate epoxide. And this is able to bind to various macromolecules and cause a lot of problems. So we initially started our study by injecting ethyl carbamate into 2-month-old AJ mice and letting these mice sit and then sacrificing them at monthly intervals up to 12 months. And here I show you a typical immunostaining pattern for TGF-beta1 ligand, as well as the type 1 and type 2 receptor proteins for TGF-beta in these lung tumors. And in the left panel, middle panel, you can see intense brown staining for TGF-beta1 ligand and type 1 receptor within all of the stages of tumorigenesis. However, if you look at the type 2 receptor in the right-hand panels, you can see that there's decreased staining for the type 2 receptor in these tumors, indicating that there's decreased production of the type 2 receptor in these tumors. This is better shown in this slide on the left, where we compared staining for the type 1 receptor with the type 2 receptor. And while we can see brown, very brown staining in the adenocarcinoma and in the normal adjacent bronchiole, we can see very reduced staining for the type 2 receptor protein in the tumors, but appropriate staining like the type 1 receptor in the normal bronchioles. We also looked at the messenger RNA for the type 1 and type 2 receptor in 5 lung tumor-derived cell lines, mouse lung tumor-derived cell lines. And I point your attention to the PCC4 cell line on the far right. This cell line was derived from an ethyl carbamate-induced mouse lung tumor, and we see decreased message for the type 1 receptor and type 2 receptor in the cell line. We also investigated the expression of the protein in messenger RNA for TGF-beta1 and type 1 and type 2 receptors, and benzpyrene-induced aging mouse lung tumors as well. And the top panel show intense staining for the TGF-beta1 ligand in the type 1 receptor, but decreased staining for the type 2 receptor. Middle panel shows also abundant expression of the messenger RNAs for these proteins, for TGF-beta1 as well as the type 1 receptor, but reduced staining for the type 2 receptor. The bottom panel show equivalent immunostaining for all 3 proteins in the normal surrounding bronchial tissues. So we're seeing in these mice as well decreased expression of the type 2 receptor mRNA and protein in these advanced tumors. So our models show that with reduced expression of the type 2 receptor, we're seeing lung tumor promotion. Now, we ask the additional question, does deletion of TGF-beta1 affect lung tumorigenesis? For this, we use the C57 Black 6 TGF-beta1 mouse. Now, the C57 Black 6 TGF-beta1 knockout mouse was generated at the NCI in the 80s. And the mouse shown in the upper the TGF-beta1 knockout mouse, in which both alleles of TGF-beta1 have been deleted, is born, and is smaller than its wild-type littermate. But there's a problem. By 21 days after birth, it starts to succumb to a general wasting phenomena and dies very quickly. So this mouse would not be a good model for use in a tumorigenesis study. But unlike the knockout mouse, the heterozygous littermates, in which only one allele of TGF-beta is deleted, survives birth, thrives, and goes on into adulthood. So in collaboration with Lola G. Wakefield at the NCI, we collaborated to first, we treated these mice with diethyl nitrosamine, which is a liver carcinogen, and asked if there was any impact on the liver tumors. And we did see that in the liver, there was increased tumorigenesis in the liver. But unexpectedly and surprisingly, we also saw increased tumorigenesis in the lung, even more elevated than in the liver. So we were kind of scratching our heads and trying to figure out what was going on. So in order to get the system into a better system for looking at the lung and not the liver, we decided to cross the C57 Black 6 TGF beta 1 heterozygous mouse with the wild type AJ mouse to make an F1 generation of TGF beta 1 heterozygous and wild type litter mace in the AJ Black 6 background. And in this slide, we did a preliminary immunohistochemical and in situ hybridization to show that there was decreased staining and expression of both the TGF beta 1 protein and message in these mice. These are, I'm showing you normal mouse bronchioles, okay, compared to the wild type litter mace. We also looked at expression of TGF beta 1 message using northern blotting, and again, the heterozygous mice show decreased levels of TGF beta 1 compared to the wild type mice. And also we did the same thing with competitive RT-PCR, showing that the heterozygous mice have contained fewer copies than the wild type litter mace. So our model was what we hoped it would be. So again, we injected these mice with ethyl carbamate, 2 months of age, and sacrificed the mice at monthly intervals. And we had, using the 2 genotypes, TGF beta 1 heterozygous and TGF beta 1 wild type. And we looked at the tumors. And this is forgive me, it's a busy slide, but the essence shows that there is increased hyperplasia and adenoma in the TGF beta 1 heterozygous mice shown in red compared to the wild type mice in green, okay? And I want you to focus more specifically on the carcinoma because it seems like it takes up to, well, in the heterozygous mice, we begin seeing carcinomas by 4 months of age, but it takes at least 12 months before we start seeing any carcinomas, and very few carcinomas in the wild type mice. So there's increased tumor incidence as well as multiplicity and decreased tumor latency in the TGF beta 1 heterozygous mouse compared to the wild type litter mate in terms of tumors. Now, we also, since we're interested in the type 2 receptor protein in these lung lesions, we stained for the type 2 receptor. And again, in the carcinomas, specifically in the carcinomas, we see reduced staining in the heterozygous mice compared to the wild type litter mates. So the TGF beta pathway is being affected in lung tumorigenesis in these cells. Now, we were interested in looking at what happens with successive tumorigenesis. So we looked at the various stages of tumorigenesis, hyperplasia, adenoma, and carcinoma, and we observed decreasing amounts of the type 2 receptor messenger RNA with increasing tumorigenesis. Our next question was, does deletion of TGF beta 1 and mutation of KRAS in combination affect lung tumorigenesis? And for this, we use the TGF beta 1 heterozygous KRAS latent activatable mouse. The KRAS latent activatable mouse requires at least one step in order to activate the tumorigenesis system. So we crossed the TGF beta 1 heterozygous mouse in C57 Black 6 with the KRAS latent activatable mouse, which was in the background SV129, to make the F1 generation of a TGF beta 1 heterozygous KRAS latent activatable double mutant, the KRAS latent activatable single mutant, the TGF beta 1 heterozygous single mutant, and the wild type litter mates. So we had 4 genotypes here to be concerned with. And we let these mice go for about 4 months, And then we opened them up and looked at their lungs. And at the top two photographs, A and B, you can see pearly white nodules on the surface of the lungs in the double mutant and the KRAS single mutant. But in the TGF-beta1 heterozygous mouse and the wild-type mouse shown in the bottom two panels, you can see no nodules. In fact, it's very rare that you saw any kind of nodule there. So we looked at the effect of TGF-beta1 gene deletion and KRAS mutation on mouse survival in these four genotypes, and we observed that the TGF-beta1 had KRAS double mutant shown in A, and the KRAS single mutant shown in B showed decreased lifespans in the double mutant and single mutants KRAS mice compared to the TGF-beta1 heterozygous mouse and the wild-type mouse on their own. We looked at the pathology of the lung lesions in these mice and observed increased hyperplasia and adenoma in the single KRAS mutant mice, but in contrast, we saw increased carcinoma in the double mutant mice. So it takes a while for the carcinomas to build up, but when they do, the double mutant really is explosive in terms of tumors. We stained for the TGF-beta1 and type 2 receptor proteins in these lung lesions, and I draw your attention to the panels on the right where you see the pink arrows, and it shows reduced expression of the TGF-beta1 ligand and the type 2 receptor in the double mutant tumors compared to the single mutant KRAS latent activatable mouse in the carcinomas. Now, we're also interested in what's happening downstream. So we looked at, in addition to the various SMAD proteins that are involved in TGF-beta signaling, and here we show a Western blot where there are expedited reduction of the type 2 receptor and increased production of the SMAD3 protein in the double mutant on the right compared to the single KRAS mutant on the left. Very little happens in the wild type or TGF-beta1 heterozygous tumors. So to make a synopsis of what's going on here, by a Western blot, the proteins for the type 2 receptor are expedited in terms of reduction, while the SMAD3 protein is expedited in terms of production. So there's a difference here. In addition, we see reduced production of both the SMAD4 and SMAD7 proteins, as well as expedited production of KRAS and RAF1 in the RAS pathway. We also used real-time RT-PCR to confirm essentially the same thing. So we're knowing that TGF-beta also can impact on apoptosis. We looked at apoptosis in the double mutant and the single mutant KRAS lung tumors and showed essentially reduced apoptosis in the double mutant adenomas shown in red compared to the KRAS single mutant. So our system shows that using these various mouse models, there's decreased expression of the type 2 receptor, which can lead to lung tumor promotion, as well as activated RAS MAP kinase can lead to lung tumor production. Decreased production of SMAD4. I really didn't show you much of this, but there is decreased SMAD4 production, as well as compromised apoptosis, also contributing to lung tumor promotion. So our system is really geared for lung tumor promotion. So the context in which TGF beta really is put into makes a large difference to what it can and cannot do. Specifically, when it can act as a tumor suppressor, when it can act as a tumor promoter. So I'd like to acknowledge the people who were involved in this work who have since moved on. Jerry Anderson, Yan Kang, Alina Namova, Joyce Pandy, and Sarah Umford. And we got we obtained the latent activatable KRAS mice from the Tyler Dex lab at MIT. So I'll entertain any questions that you might have at this time. Okay, thank you. Yes. Mm-hmm.
What does it look like when you stay inactive? You have just as much present?
No, because the inactive cannot really participate in tumorigenesis. It has to be activated. Well, no, the inactive is not secreted. It has to be activated to be secreted. There are a number of compounds that can activate it, such as acid. Decreased pH can activate TGF beta. And it's not really understood what really activates it internally in the body. Yeah, still being looked at. Yes?
Is there any other kind of cancer in other organs other than the liver and the lung? TGF beta knockout mice increase the rate of the other organs?
Well, in our systems, we treated it with a liver carcinogen and a lung carcinogen. So that's why we got the liver and the lung, basically. I suppose it can be turned on by other types of tissue promoters as well, systems.
Is there any relationship between the TGF beta and then the lifestyle and the effect of the
That's a good question. I don't think it's been looked at. I would suspect that there probably is, depending on what the environment is. You know, I think there would be.
Okay, that'll do it.
Okay, thank you.